And welcome, everybody. Welcome to Ask a Geologist. We are today, it's my honor today to introduce our speaker, Elizabeth Padilla Crespo. She's a dear friend of mine, and we went together to college. She was at the University of Puerto Rico in Mayagüez. And from there, she went to Georgia State Institute for her master's degree, following a PhD from the University of Tennessee at Knoxville. And currently, she's at the Inter-American University at Aguadilla. She's a distinguished professor and coordinator for student internships. And it's our pleasure to have her today to talk about a known passion of hers, which are microbes. So with further ado, we open the floor to Dr. Elizabeth Padilla Crespo. Thank you so much for being here today.
Thank you guys for the invitation. I am very excited to be here with you today. And I would like to extend lots of thanks for the team behind this event that I know they put so much heart and work into producing these phenomenal seminars. Like Patricia mentioned, I'm going to share with you a little bit about my passion, which are microbes, the unseen majority. A little bit about myself so that you get to know me better. I was born and raised in Puerto Rico, a beautiful island in the Caribbean. I have degrees in biotechnology, microbiology, and biology. And after I went to college with Patricia Irizarry, I went off to live in Georgia, then Tennessee, DC, and now I am back to Puerto Rico. I've been here since 2014, I believe. I would like to share that I'm a first generation student, proud to be from the public school system. So for those students out there that share a similar background, know that there are no barriers and that you can always accomplish with hard work and believing from your heart, you can accomplish everything that you want. So I have been involved in various projects that are interdisciplinary. Sometimes I've been working with things that are related more to chemistry, even to engineering or environmental scientists. But between all of those projects, more than anything, I am a microbiologist. And this is why I want to share with you guys the fabulous world of microbes. Here you see in this picture, I like to work in the laboratory. And early on, I was very lucky to be involved in research and projects that kind of led to where I am right now, which is I'm a researcher and a professor at the university. So also I want to encourage you students, what you're doing today is part of your professional development and part of the extracurricular activities that you can do to cherish and expand your horizons. So it's very good that you're here taking the time to be part of these seminars and to know a little bit more about science in general. So why do I love microbiology? Because it's fascinating. It's life at the micro scale that you cannot see with your bare eyes. And there's a lot of diversity, different lifestyles, different forms of living organisms that is just huge. And we will talk a little bit about this, but also within the realm of microbiology, we not only study living things like bacteria, we also study viruses like you see here. And because of the current pandemic times, we've got to know a lot more about viruses and how they affect life, especially public health in our case during these COVID times. So I encourage you to, if you're passionate about it or curious about to know more about microbiology, to go and read more about this fascinating field because it's really is a great venue. So why do microbes rule? Well, they are everywhere. They're omnipresent in nature. They're found in soil, in the air you breathe, in the tap water that you drink. These mediums are not sterile. Microbes are everywhere. Actually, in the food also that you eat, and I will talk about that later. Here, what you can see is a picture that became famous of the microorganisms harbored in a little boy's hand. So its mom, he was playing outside, and when he came inside, she pressed his hand against this agar, which is a solid medium where you can grow microorganisms. And after 24 hours, these appear. All of these type of bacteria with different colors, they mean that they're different species and different lifestyles that are harbored in your hands. So even if you think that you clean your hands very well, microbes are still there. And they rule because they encompass most of life here on Earth. They make 98% of the biomass of the oceans. So you think about it, we're talking about the weight. So you may think about the ocean and think about huge whales, sharks, algae, but actually 98% is like almost 100% of the world's oceans biomass are microbes. In 1 gram of soil, which is almost like a teaspoon of dirt, right, from your backyard, there's 40 million bacterial cells in it. You just don't think about it usually. In a milliliter of fresh water, like if there's a creek right by your house or in your neighborhood, in that creek, a milliliter of water, which is this much, not very, there's a million bacterial cells, and the water may look to you transparent, but they're there. And also, like I was saying, they're a big part of commercial applications, and also things that we use in this case for us to eat. Sauerkraut, yogurt, wine, cheese, all of those things are products of microbial activity. There's microbes that were used in the process so that we can have this delicious food for ourselves. There are a lot of things that are fermented, and that's the word we use for this when there's microbial activity going on some substrate. And when there's that fermentation goes on, there's also a product that is sometimes something delicious that we used in our for us. And lastly, and I like to have these numbers so that you can have an idea of the magnitude that we're talking about. There is an estimated 5 million trillion bacterial cells on Earth, with a total biomass equaling that of plants and animals. So if we think that our planet is mostly filled with trees, vegetation, animals, humans, you're wrong. There is equal biomass, so equal weight, equal amount of these than microbes. So this is why I call them the unseen majority. They are this huge force of nature out there that we sometimes don't think about. There are more species of bacteria in this planet than any other form of life. As you can see this here in this graph, bacteria represent 78% of all species in our biosphere on Earth, while plants are a little bit, fungi, animal, and protists. So with this, I'm giving you numbers and some idea of how big force of nature they are. We biologists in general have studied life, and we like to classify things. And what we have gathered information, mainly from genetic sources, is that all life on Earth are from 3 different groups, big groups, that we call domains. And I will shed light into this in the next slide, but just so that you know, these 3 domains are bacteria, archaea, and eukaryotes. So everything that has life on Earth is part of one of 3 big groups called domains. And this is what you can see here. This is a tree of life, even though it's kind of on its side. And the 3 big domains are represented here. So what I want to show you is that everything that is here on pale yellow is microbial life. Eukaryotes, which are this on this region, is what harbors the big animals and organisms. So trees, us, we are here on this blue part. But part of the eukaryotes are also microscopic, and that's where we have yeast, protozoans, and little animals that you cannot see with your bare naked eye. And there's this huge domain that is bacteria that is full of diversity and that we sometimes associate with disease more often than anything else. And lastly, the domain archaea, also microscopic, also a single, so there are single cell organisms. And they're like kind of old bacteria. This is why they are called archaea. They're rustic. We think they're probably one of the first, they reassemble one of the first forms of life on Earth, and they live on extreme environments. So the big message here is that most life on Earth is microbial. So if you're a microbiologist, you can think of yourself as an expert in life, and you will always have a job out there if you get a degree related to this field, because like I'm telling you, there's a lot of opportunities and things to study. Within the realm of microbiology, I am an environmental microbiologist. And sometimes I have studied things related to geological processes. So I have been able to enter the field of geomicrobiology. What I like about being an environmental microbiologist is that I can work in the lab if I want, like you saw some pictures that I show you, but I also get to go to the field, to the sites, to nature, and collect samples, work with dirt, with water, and take samples so that I can bring back to the lab and do my analysis. Two of the areas that I've been looking at are extreme environments and bioremediation. These are two main topics that I have focused my research and my work that I will explain to you in the next few slides. When we talk about extreme environments, we're talking about places with high temperature, toxic chemicals, and overall harsh conditions where we as humans cannot live and where we think maybe there's no forms of life. But actually, microbes can thrive in these environments also. What you see here are different places on Earth that are extreme. For example, glaciers, ice formations, there's volcanoes, hydrothermal vents, the deserts, places where the pH is very low, very acidic, where a higher organisms cannot live because it would just die. The depths of the ocean where there's no light, where the pressure is high or the temperature is super low. These extreme environments, we may think life doesn't thrive there, but actually microbes can make a living at these sites because they have been here on this earth longer than we have, 3.5 billion years ago, and they have been very clever into developing strategies to live in these environments. Studying this type of bacteria that thrive in these environments is very interesting because that's a link to astrobiology. And what does that mean? Well, it means two things. One, if we study these microorganisms here on Earth that thrive on these very harsh conditions, we maybe get an idea of how life might be outside of our planet. For example, Europa. Europa is one of the moons of Jupiter. It's thought to have liquid water. So it's one of the places we're looking at to see if there have been, or could be life. So, and if there is life, or there could be, it will be probably microbial, and it will be similar maybe to one of these microbes that we study on Earth in Yellowstone hot springs, for example, or in the desert of Atacama, which is is a big desert that resembles a little bit of the Martian environment. And this desert is in South America. So like I'm saying, extremophiles, they can tolerate temperatures up to more than 100 degrees. So boiling water is 100 degrees. So they tolerate temperatures higher than this. pH that is very low or high, so very acidic or basic conditions, and even radiations. There's a bacteria called Deinococcus radiodurans who just can survive high amounts of radiation. So, and not only do studying these microorganisms here can tell us about astrobiology and life elsewhere in the universe, they may tell us also something about the origin of life here on Earth. How was the primitive Earth here before the dinosaurs, before we humans and higher organisms came? We can know about these clues, a little bit more about this by studying these fascinating microbes on Earth. So I told you a little bit about my interest in this weird type of bacteria, the one that thrives on extreme environments. And now I will tell you a little bit about another area that I am interested in, which is bioremediation. Bioremediation is the use of naturally occurring bacteria that can help us restore contaminated sites. So how can we do that? Well, these microbes just have the capacity naturally to degrade toxic compounds. They derive energy from what causes harm to us. So a chemical can be carcinogenic. It can cause us cancer. But a bacteria can use it for food, for carbon, as a source of energy, etc. And microbiologists like me have been studying microbes that can degrade contaminant in a variety of environments. And that can be soil, water, sediments, or the open ocean. And here you have a bacteria, it's sitting on the table. It's a cartoon waiting for a toxic compound just to come in, so he will eat it. But that is actually what happens when there's a problem. And let's think about a spill of a hydrocarbon spill. It can be gasoline, oil, diesel, jet fuel. When these accidents happen, it's an environmental disaster. And our main thing should be, to fix it and to clean it so that it doesn't cause us more harm to the environment. It's a big ecological disturbance when these type of accidents happen. But the thing is, we have a solution, and we study this in the laboratory and in the field to provide answers to these problems. And you see again here, in the case of hydrocarbons, the microorganisms become close in contact with the oil, and it could be a variety of hydrocarbons, different type of hydrocarbons. And then it takes them as a carbon source, it digests them, and the product will be, in this case, CO2 and water. And that's how you can clean up contaminated sites that have been impacted, in this case, with oil spills. So I talked to you about the two main projects and areas that I work on: extreme environments and bioremediation. And the next few slides, I will show you pictures of me doing the field work when I have gotten the opportunity to sample and visit these scenarios that are very interesting. Here you will see I am in Azores. Azores is an island off the coast of Portugal that is volcanic. There are more than one volcano in the area, and there's this hydrothermal vents and fumaroles and hot springs close to the volcanic area. I got the opportunity to go here in 2016, I believe, as part of a group of geologists, geochemists, and microbiologists that got together to go there and sample. And I'm very fortunate that I was part of that group. Very international group, which is great because I love diversity, and science is all about international collaborations. Here I am close to a fumarole, which is one of these formations. There's gases, hot water coming in, and we were sampling for sediments and also water to take later back to the laboratory and make microbiological and genomic analysis. Here is a little map where you can see Azores is off the coast of Portugal, and I like to make the connection that I went from one island to another island to represent a Latina and women on science, because we definitely need more representation in the type of work that we do. Since I know there's geology aficionados out there, one of the sampling events took place close to this beach in South Roque, Azores, Portugal, where the sand is black, completely black. And although I did not take samples for my purposes here, I was just astonished and amazed about being, for me, the first time in a beach coast where the sand was completely black. And again, this is because of the volcanic activity nearby. I also have gotten the opportunity to go sampling in research vessels. And in this case, you will see here, this is a tramp that we place in the bottom of the ocean to take sediment and also benthic organisms, big and small. And of course, I always take samples for microbes that I take to the laboratory to study. This was done in Woodsville Oceanographic Institution out of the East Coast in the US, one of the finest places to study oceanography and microbes related to the ocean. This may be the last research journey that I will share with you guys. It took place when I was still in graduate school in Tennessee. In this expedition, we got the opportunity to go to Sweden, to another island, which is called Aspo. And this is very particular because in this island, there is a tunnel that goes hundreds of meters below the subsea floor. And I got the opportunity to go for 450 meters below the subsea floor. And you see there's a minus sign here because it was below, and it was like a journey to the center of the earth. You can see like the pressure changing. It got hot as we were very deep. So it was a very unique opportunity. To give you an idea, the Eiffel Tower is 300 meters. So we descended a distance longer than the Eiffel Tower. And what we did there was what I do in other places. I go sampling. And this is the type of formations you see down there. In these biofilms, and that's how we call this aggregation of microbes, had a brownish, reddish color, and that's because they oxidize iron. So there's bacteria that can use metals, minerals as an energy source, and they can transform them. Like I'm telling you, they're very clever. They will use anything they use, they can, to gain energy and to live. So this is the type of sampling we did. They're very interesting. I've never seen an environment like this again. And like I mentioned, yes, so there's bacteria that can use these metals and minerals as a source of energy. In the last maybe 20 years, we have gained a lot of knowledge about how they can do this. And you see here, this is two of the mainstream news where they say, you know, bacteria that can breathe rock, that can eat rock, etc. So the future for in this field, like I'm telling you, microbiology is super exciting. There's a lot to do. I told you a little bit about bioremediation and about astrobiology, and I will touch down on other applications so that if you're interested, you can go and explore more about it. Bacteria can serve as a natural fertilizer, as a biofertilizer to improve plant growth. And how do they do that? Well, you have probably seen your neighbor or your mom or somebody fertilizing a plant. So when they do that, they're adding nitrogen, phosphate, and potassium, and nutrients that the plant needs to grow. Nutrients that sometimes there are not enough of them in the soil. So these fertilizers are synthetic, and there's some concerns about too much use and the cost also of synthetic fertilizers. And also, if you want to go organic, you cannot use a synthetic fertilizer. Well, the thing is, there's bacteria out there who naturally interact with the plants and provide the plants with these nutrients. So there's a synergy, a cooperation between the bacterial groups in the soil and the plants so that they can mutually benefit of this exchange. In the case of a certain type of bacteria, they can fix nitrogen. So take the nitrogen that is in the air and fix it, make it available so that the plant can grow. And that to me is amazing because you're talking nitrogen that is here, that I'm breathing right now, in the air, bacteria can trap it, and then under the soil and the roots of these plants, provide it so that the plant can use it. And in the future, and I mean, as I speak, there's these products out there who are just little seeds that are coated with bacteria so that you can provide it to your plants. And in an agricultural plantation that is big, this can provide a lot of benefit. In the terms of bioremediation, what we are doing, well, we are studying bacteria that can transform and degrade very strange compounds. For example, there's bacteria that can transform compounds that have chlorine. So very toxic compounds. And they can use it as an electron acceptor. And what does that mean? Well, in aerobic respiration, if you can see here, Homer, oxygen is our ultimate electron acceptor. And we breathe oxygen and expel CO2. But there's bacteria that, analogous to this process, breathe these compounds. They take it as an ultimate electron acceptor. And in that, we call that organohalide respiration. In that process, the compound that is toxic gets transformed, and the chlorine atom gets removed, and that toxic compound is no longer toxic. It becomes something that is benign, that again, it doesn't represent a public health concern, and it's not harmful to the ecosystem, for the plants, animals, and for us as a society. This part of work is the one I did my doctorate in, my PhD. I helped assign function to a gene, the genomic component of a bacteria that was implicated in the transformation of a compound that is harmful, a compound that was used in the past as a pesticide, that is found in various places in the US and in the world, sadly. Well, I helped describe the type of bacteria and microorganisms that can take this compound to this benign form. And that way we can clean and restore these contaminated sites. An area that is now growing very rapidly and there's a lot of interest is the microbial degradation of plastics. And you can see here, water bottles and plastic that is not reusable is problem of the first world. What the good thing at least is there's bacteria, microorganisms that have been proven to be able to degrade it, and we're studying how they do it. We don't know a lot of it yet, and how we can better improve these processes so that we can help the environment. Lastly, on the terms of bioremediation, like I mentioned, there's bacteria that can eat radiation. And what I mean by that, there's microbes that can transform uranium, for example, radionuclides. So places that have been contaminated in the past by use of nuclear waste can be treated with this bacteria under the soil or in the water that can transform uranium and make it at least so that it's not more harmful to the environment. And this is a bacteria, Geobacter, that is involved in this type of processes. It's the star of uranium bioremediation. And also, if you like more things that are not so much environmentally related, if you like also public health and medicine, you should know that your body is also an environment. Your body is only 10% human. And what I mean by that is that you're mostly microbes. There you have microbes in your skin, in your hair, and most of them in your intestinal tract. That they help you in digestion. They produce vitamins for you that you cannot synthesize, and they promote the health of your immune system and make you just a stronger human being. As you can see here, this is epithelial cells, a human cell, the blue big thing here. And the darker, smaller points is bacteria, so that you have an idea of the scale. You're mostly bacteria. You're less human than what you think. And when we think about ratios, we say there's 10 bacterial cells for one human cell that you have in your body. So although we think about microbes as disease-causing organism, and we want to clean our hands so that we don't get sick and so on, that is important. But most of the microbes out there are actually, and the ones that you have in your body, they are not harmful, they're actually your friends. And why do we care about all of this field of microbiology, all of these applications, and me specifically, why do I keep on working on the area, is that we know very little. There's an untapped diversity. There's a universe of microbes out there waiting to be discovered. And this is what I try to portray in this slide. What we know is very little, and there's an estimate that we know less than 1% of the microbes that live out there. And that's huge. It means that 99% of the microbes that live in this planet, we know nothing about. And how can that be? Well, most of them, we cannot culture them in the laboratory because, as you can see, they like very specific ways of living, ways that we don't know about. And sometimes doing the easy plating in the lab for them, giving them all the nutrients, water, they don't like that. Sometimes they really like these very harsh conditions, and we cannot just grow them in the lab and study them. So remember this, 99% of the biodiversity, of the microbial biodiversity is unknown. So we need more microbiologists out there, especially the ones that like to go to the environment and sample to know more about these very interesting life forms. So what I do now is, like I told you, I go to the field, sample, and I bring them back to the lab and work with my students. At the Inter-American University of Puerto Rico, I like to provide them with hands-on opportunities so that they can play with their hands and get excited about this field. And with that, I am open to questions. Thank you.
Elizabeth, that was wonderful. We have quite a few questions in the document for you.
Okay, I'm looking for it. Where is it? Can you provide me with the link again? I think I closed my tab.
I just sent it to you in the chat.
Oh, thank you. It's opening. Yes. So, so Mary from Scotch Plains just asked something I mentioned about, are we still finding new kinds of microbes? And the answer is yes. Like I mentioned, 99% of the diversity out there, we don't know it. There's papers and research going on as we speak, and almost every week, I would say, there's a new microbe being described. like I mentioned, some of them eat rock. They are very clever in how they get energy. Something like iron, for example, pyrite, which is called also fool's gold, is um, is a great source of energy from some of these microbes. This was a very interesting question. Do microbes share any DNA with us? Well, I can tell you that there's been evidence that we in our human genome, and that was the project, we have in our genome bacteria genes, and that is amazing. And you can think about, like I told you, we harbor all these type of communities in our bodies, and we are so in contact with them that at some point we have shared DNA with them. And at least I cannot tell you there's a human gene inside a bacteria by nature, but we humans harbor DNA genes. It is estimated that more than 100 genes in our genome come from bacteria, and that is data that researchers have looked into, and every day things change. So maybe in the future we will know more about them. The earliest microbe, well, at least we know the earliest fossil are extramatolites. And I didn't talk about them, but this is the formations of limestone produced by cyanobacteria in the primitive earth. So around 3.5 billion years ago, when life first appeared, we have evidence that it was unicellular, microbial, and then at least some of the first type of life were photosynthetic bacteria, cyanobacteria. These bacteria, as part of the process, just like a plant, gain energy from light and produce oxygen as a byproduct. And actually, the transformation of Earth to be habitable, to be an environment for higher organisms, happened because of the cyanobacteria. Cyanobacteria, by breathing, produce the oxygen that made the atmosphere oxidize. This event is called the Great Oxidation event, and it opened the way so that we higher living. How do we acquire bacteria before we are born? This is very interesting, and I am an environmental microbiologist, so I know less about the human and microbe interactions, but it was thought that we were first colonized by bacteria at childbirth. And there's evidence that those who are born by C-section have a very different composition of microbes and also of pathologies of maybe sickness once they're older than those that were born by natural birth. And it's because they came in contact with different populations of bacteria. I have also read that we thought that when we were in the womb, it was a somehow sterile environment without bacteria, but I have read documents and research that points out that maybe our interaction with microbes may even have started in the womb, in our bellies, in the mom's belly, to say it like that. I haven't discovered a new species of microbe, but this is one of the things I would like. Here in Puerto Rico, we have a lot of very interesting environments. We have our own hot springs. We have a desert-like environment in the south of the island. The mountains, which are more template in temperature. So I would definitely like to have the opportunity to isolate and know more about microbes here in Puerto Rico and have the opportunity to name them maybe and describe them. This is a very interesting question. Ria's mom would like to know do hurricanes have on microbes living in the environment in Puerto Rico? Well, I will tell you from experience, a hurricane is just a big, huge disturbance on the environment, from us humans to the plants to just the ecosystem overall, the geography of the island. But I will take this question to add a little bit more. Microbes have been linked to cloud formation. And that is to me, that's almost like science fiction. And I am not an atmospheric scientist, but particles that are suspended in the atmosphere, in the stratosphere, sometimes these particles is where the condensation starts to happen to make up clouds. So a microbe can serve as a particle where a cloud start forming. And there are scientists in my area, to answer all these very interesting questions. Also in Puerto Rico, we get the Sahara dust, which recently I saw that the quality of the air was not very good and I'm very allergic. It's because we get part of the Sahara desert dust as a cloud here in Puerto Rico. So people have studied this and have linked certain type of microbes that have come with the Sahara dust cloud. And sometimes people say maybe they're frequent flyers, maybe they're cosmopolitan because they are going from one place to the other, and maybe they can be related to disease, to asthma, and so on. So people are studying microbes that are linked to clouds and to atmospheric events. Let me see. What kind of bacteria before we are more? If I could go anywhere in the world, where would you go to sample? I would be very intrigued to go to the North Pole and at the glaciers, because I know people take core samples from glaciers, from ice, and it's very intriguing how they find microbial life inside those cores. But I think it will be very difficult expedition for me, specifically dealing with the low temperatures and all the year that I would need to have. But I know of colleagues that do this in the research and they love it. I would really like to go to the Tacoma Desert, that one that I told you that has a topography similar to Mars, and that is in South America. And my research and my travels have just not taken me to South America much. So I would definitely like to go there and take samples and use them to know more about this area of astrobiology that I am also interested in. Other places that I have been to sample. So I was able to go to New Zealand and study the ecology of different aquatic organisms there. And more recently, Thailand. So that picture that I show you very at the end, where I'm both at the field and that was actually in Thailand, in Bangkok. And we were studying photosynthetic bacteria. So I got the opportunity to go to these ponds, lakes, and sample the biofilms. You saw all those green things, and water and sediments from these ponds. I got the opportunity to go there with 10 more students from the US, 3 of them from Puerto Rico. And it was a very unique experience because more than the science, it was also a cultural enriching experience, us doing research with the professionals there in the university and finding common ground between all of our differences, language, style of food, cultural backgrounds, but finding this common language of science that unified us. And at the end of the day, you could see, you know, we are not that different at all. And we were driven by the same passion and asking the same questions about life on Earth and about these intriguing things that we want to answer regarding environmental microbiology. Christopher would like to know, what do different microbes provide to other organisms? Well, like in the case of the biofertilizers, they can provide food, microbe to a plant. They can also provide protection. Maybe the microbe is producing a compound that is that protects them from another pathogen that may infect the plant. So usually we think about microbes sometimes as agents of infection, things that cause pathogenicity. But like I mentioned, we have to look at it on the other side also. Microbes protect us. And in that case, that have been shown in plants. They make these synergies and relationship with microbes and protect them from others that do cause them disease. To follow the same line of thought, when I say that they provide the plant with food, I'm saying things like phosphate, like nitrogen. They also provide vitamins and hormones so that the plant can grow. Let me see what else I see in the question. Okay, so Christopher would like to know if microbes can live in these extreme environments and are asexual. How do they reproduce? And can they pass on their genes to future offspring? So yes, bacteria are asexual. They divide by binary fusion. One cell divides and ends up having an offspring. But how do they do it? Sometimes when they are in the extreme environments, the metabolism rates on them is very low. Sometimes because they don't have a lot of nutrients, carbon source. So they cannot go, they cannot be like a runner in a marathon running very fast because you will run out of energy. So when they don't have a lot of energy source, the metabolism just slowers, and reproduction also slowers. So that is how they make a living, and sometimes they just don't, and they are just, the cell lingers. It becomes a spore sometimes where there's no humidity and water levels are very low. There's bacteria, like the whole cell becomes endospore. And like that, it protects itself from being in this desiccated way because there's no water available, but it maintains its integrity at least. And whenever water comes, she can bloom again and live at the same rate that she was doing before. And of course, when they reproduce, it means that their genetic information is also going to the offspring cell. Oh. Okay, how would microbes treat pollution from the nuclear waste, from places like nuclear waste or plastic. I didn't explain very well how bacteria can transform uranium. So I will take a minute or so to explain it. Uranium is an element. It's in your elements table. So you cannot degrade uranium. It's like you cannot degrade carbon or hydrogen. It's an element. So how is it that I'm telling you that bacteria can bioremediate places with nuclear waste, if the nuclear waste is uranium? So what happens is that there's a form of uranium that is soluble in water. So it means that it can move in water. And we are talking groundwater. So all of these contaminants, people throw them in the soil and they end up in the groundwater below the surface. And so in this uranium is in that form, it can move in that groundwater, and that's a threat because it can reach a creek, a river, a drinking water, a place for the family, maybe if they're taking groundwater as a source, it can reach the ocean. So what happens is bacteria that can transform this uranium that is soluble, which is uranium-4, to uranium-6. So uranium-6 precipitates. It becomes like a powder, like a dust. So at least you can contain it. So when people bioremediating places that are contaminated with uranium in groundwater, they promote that the bacteria will turn uranium-4 to 6, which then precipitates, and at least you can contain it at the site. That is how it's done in that case. The scientists have to collect the microbes and bring them to the site of pollution? That's a very interesting question. And so in bioremediation, there are two practices. One is called biostimulation, and the other one is bioaugmentation. If I want to restore a contaminated site with biostimulation, I need the bacteria that can do the job for me. I need it to be already there. So I just stimulate the bacterial growth. And that can be, I can add food to the place where the bioremediation is going on. I can add air, oxygen if it's needed, provide all the nutrients and conditions so that the bacteria in that soil can grow, reproduce, be happy, and contaminate and degrade the contaminants for me. So that is called biostimulation. But there are some sites that's just a naturally occurring community, bacterial community doesn't have the bacteria that can do the transformation. They're just not there. And in that case, and I think that's what that question is alluding to, I can take the microbes that I have in the laboratory, grow them in big vessels, in big bottles, to the environmental site, and dump them in the ground so that they can grow and do the transformations for me. Just like I can inoculate a Petri dish or a tube of culture in the laboratory, I can do that with with a site that is contaminated. So that is one of the applications that I enjoy the most when I do bioremediation, that I get to explain these techniques that we can use. Do I have
There's
more
one.
question?
It is, Tim would like to know, would it be correct to say that the organs that make up humans, from skin to brain and beyond, are the human part of us?
That's an interesting question. Would it be from skin to brain are the human part of us? Yeah, I mean, our organs are made of tissue, which are made of human cells. But like I'm saying, the perspective that I always like to remind or bring up is that you harbor in your body more microbes than human cells. You actually have more bacterial DNA in your body than you have human genes, just because you have much more bacterial and microbial cells. One thing that he mentions their team, he talks about the brain. And from what I know, and again, I'm an environmental microbiologist who goes to play with dirt whenever there's a chance. I don't know a lot of human microbe interactions, but I will tell you that for a long time, it was thought that the brain, one of those places that there was no microbes, no bacteria, there was an organ that you don't think it can be infected with cells because it would be very harmful for the human being. But actually, there's been brain sections where they have identified what it seems to be, colonization of bacteria from a person that wasn't sick, It was not somebody that had meningitis or anything else. So now we're thinking twice about saying there's places on our body where there's no bacteria, like maybe the brain. They're being very careful about doing that. The thing is, it's a hard question to answer because what if the bacteria or the microbial cell that they see, maybe that was a contamination of the process of taking the sample, prepare it, and then you actually looking at it. So there's always that question about maybe it was not there in the beginning, but you contaminated the sample, and that's what you're seeing. But at least there's data that indicates that know that even our brain can harbor bacteria in healthy individuals. Of course, the numbers would be very low because otherwise it would be an infection, and then it wouldn't be good.
Thank you so much. I think you answered all of the questions, which there were a lot. So thank you again for taking the time and doing this presentation for us. It was very interesting. And thank you to our audience for joining us today. Our next Ask a Geologist will be on Friday, May 14th at 1 p.m. We will have Dr. Rosana Grafal Soto, who is an associate professor at the University of Puerto Rico, Calle, and she'll be speaking about coastal dunes. But we do have an event coming up before that, a lot sooner. We have our virtual meteorites late night, and we will have a guest presentation about meteorites by Dr. Julianne Gross, who is an associate professor at Rutgers and a NASA deputy curator of the Apollo moon samples. She'll be speaking about meteorites and her experiences discovering them and studying them. And we will also have trivia, and we will have an activity for that late night, which will be at 6 p.m. on May 5th. Okay, so that's Wednesday, May 5th, our late night, and then we will have our next Ask a Geologist on Friday, May 14th. So mark your calendars, and we hope to see everybody there. And thank you once again, Elizabeth, and we hope you have a great weekend. Thank you.
Thank you. Bye.